Now we're going to proceed to talk about the last few topics in Chapter 17, starting by talking about the genetic code. As we explained previously, during the process of translation, the mRNA is going to be read by looking at the codons, okay? So codons are going to be specifically found in the mRNA, and they're going to be 3-letter codes that during the process of translation, they're going to give rise to a particular amino acid. So understand that the total number of codons that exist in the genetic code are 64. Okay? So remember, I mentioned that they are triplet combinations of A, G, C, and U, because remember that we're using in the process of translation as a template mRNA. Within this 64 codons, we're going to have at least a few codons that represent a stop. Remember that that is where the release factor binds in order to release the protein that has been synthesized. And the stop codons refer to UGA, UAA, and UAG. Okay? Now, understand that these codons are utilized and they are organized in a table. Understand that you do not have to memorize the genetic code. Maybe there's people out there that would like to do that. I personally do not even have to memorize myself. So understand that for the final, you must have the genetic code table handy. Meaning that you should have it around because there will be some questions about codons and even the process of translation. So one of the things that I want to point out is that as we saw in the process of translation, AUG is the first one that is incorporated onto most proteins because that is called the start site. When it comes to the amino acids, the other amino acid that only has one codon is going to be tryptophan. In this genetic code table, remember, amino acids have a full name, 3-letter code, 1-letter code. So here we have the 3-letter code for the amino acids. So one of the things that it is known about the genetic code is that the genetic code is degenerate. And degenerate means that there are, for most amino acids, have several codons that are present. Okay? So here we have an overall picture of the genetic code, and please, for the final exam, make sure that you have this table with you in order to answer certain questions in the exam. Now, we are now going to move on to talk about genetic mutations. Okay? So understand that genetic mutations can happen during the process of DNA replication. They can happen in transcription, and rarely they happen in translation. So most of them happen during the replication or transcription. Now, when it comes to genetic mutations, understand that you guys should be familiar with the different types of mutations that are outlined in this slide. So when it comes to this, understand that these genetic mutations, okay, the types that you guys need to be familiar with are, first, substitutions or point mutations. Now, when we have a substitution or a point mutation, that is going to be specifically the replacement of one base in the template strand of the DNA for another. Now, because remember, as I just mentioned, that let's say that a base is exchanged. When this happens, again, that can result in the coding of different amino acids, but if it results in no change in the amino acid sequence, then that is known as a silent mutation. Okay? So, when it comes to point mutations, we can also have a nonsense mutation and a missense mutation. A nonsense mutation is where the base that is changed results in a stop. When it comes to a missense mutation, we can have two types: conservative or non-conservative. So the point mutation can be changing the amino acid in the conservative way, meaning we're going to have an amino acid from the same family. And what I mean with the same family is that if we compared arginine and lysine, they are both basic amino acids. If we have a non-conservative missense, we're going to be mutating to amino acids that are from a different family. So as you can see, if the no mutation at the level of DNA encodes for lysine in a non-conservative missense mutation, now we are going to have the synthesis of tyrosine. And tyrosine, remember that that is a polar amino acid. Let me just highlight that in a different color to make it easier. So when we compare tyrosine to the original amino acid that's there, now we have the exchange from a basic amino acid to a polar amino acid. In addition to substitutions or point mutations, we can always have a frame shift mutation. And understand that frame shift mutations are always going to be affecting the protein, okay? So when we have a frame shift, that is going to come from deleting a few codons or adding new codons, okay? So as you can see here, in the example that we have in the bottom of the figure, we have the normal sequence. Codon 1, CAG, codon 2, CCC, codon 3, ACT. That encodes for, and again, when it comes to this, you guys need to have your amino acids in front of you. So this is glutamine, proline, and threonine. Now, in this insertion frame shift, we are adding a new nucleotide to the sequence. So when we are adding this new nucleotide, understand that now the way that we read it in 333s is going to be shift. And what this is going to happen is that now the first codon is going to be the same. It's going to be that GLN or glutamine. But the incorporation of that T makes a brand new amino acid. So the rest of the molecule now is not going to be correct. Understand that genetic mutations occur and they can be very detrimental, but at times mutations are utilized in the laboratory in order to study specific proteins, okay? Now, when it comes to examples of genetic mutations, understand that most of us, if we are a fan of X-Men, we've seen how genetic mutations are shown in movies. But if you look at the slides, genetic mutations include, or examples of genetic mutations include, not only X-Men, which that's just fictional, it's just based on the movies, but real examples of genetic mutations include albinism, blue eyes, and having 6 digits, meaning having an extra finger. The next thing that we're going to talk about is viruses, okay? And in these times with the pandemic, understanding viruses, it is very important. Understand that viruses are not an organism. Viruses are known as particles, okay? And inside of them, they have from 3 to 200 genes. The reason why they are not considered an organism is because they require a host cell in order to replicate. So they do not have all the components for transcription and translation. That's why they invade a host cell, utilizes the machinery that we explained in the process of transcription and translation previously from the host cell in order to replicate its DNA. So depending on the virus that we're dealing with, they contain nucleic acid. They can contain DNA or they can contain RNA, okay? And this nucleic acid is contained in something that is called a protein coat. And as you can see here in the figure, we have different types of viruses. They kind of look like very science fiction animals, but that's the reality of how they look like. And if we relate it to the coronavirus, remember that if we see it, coronavirus looks a little bit on the envelope form. Now, understand, as I just mentioned, that they do not have the necessary material like nucleotides, enzymes to make proteins and grow. So that's why they need to invade a host cell. Okay? So in order to replicate, they invade the host cell and it takes over the machinery, as I just been mentioning, in order for themselves to undergo transcription and translation. So viruses is something that it is affecting the world at this moment. And as we know, the coronavirus is infecting and killing many populations around the world. But that is not the only virus that we're exposed to. As you can see, things from the common cold, influenza, warts, herpes, human papillomavirus, all of them are actually caused by viral infections. Understand that to date, we do not have the cure for any viral infection. Okay? Right now, as you guys have heard in the news, there's new treatments that are trying to be developed to deal with the coronavirus. But in general, we do not have cures for any of the ones that we have in the list that is outlined in table 17.9. The only thing that it is available for some of them, and some of us actually have been vaccinated against, like for example, hepatitis or chickenpox. Vaccines have been created in order to introduce to the body these viruses that then the body can attack and make antibodies against. So that's why in the news you have heard that they're really trying to push for a vaccine for the coronavirus, because that's the only way that we know how to treat viruses to date.